Hello, this is the Douglas Scientific Capstone Project with the NDSU, and this is the final results of the semester that the students have produced. Here we have the software to work with data produced by the array tape technologies for high throughput data and protein analysis. You can do qualitative and quantitative graphing. I'll demonstrate both here. First we'll do qualitative. The first thing you'll need to do is choose the files you'd like to work with. So you can choose the file, import it, and we can just show that you can do multiple files here. And then once you have the files imported, you can decide what you'd consider a hit or containing the protein, or on the other hand, not having the protein. So if it's above this, it has it. If it's below it, it doesn't have it. Once we have the qualitative threshold, you can select the file you'd like to visualize, and this will plot the data, and you can see the threshold line here in blue, and then you can hover over any of the plots so you can see the values on the left. And then you can also see the wells as an array tape formation, so you can visualize it that way, and you can see which ones was the hit as in red and in green, and what was a miss in the red. And that can be exported so that you can see and use that data in a different kind of file of your choice. So tab separated values, text, Excel, and comma separated values. We'll just use comma separated values for now. And for the other type of curve, to use that, you'll need to make an S-curve first. S-curves are the standard plot that you'd like to test your data against. And so what you'll need to do is choose a file that you'd like to base the standard S-curve off of, and you can import that. Once you've imported, you can select it, and you'll see a well by well basis of the values. Leave it blank if you don't know the concentration of your control well, or if it isn't a control well. And if it is a control well and you know the values, you input it wherever those wells may be. And so I've made a control curve here, so we'll just use that, and I'll just make a dummy curve right now for demonstrative purposes here. And so you can import the values as you go. And this will just take a moment, and since if you know a lot of control wells here, it could take some time, and so we've had it so you can save them over here and reuse them later, so it doesn't take you as much time to control or to test data after data. And so you can either select a 4-parameter logistics or a 5-parameter logistics. Once you've selected that and you've imported the values, you can name the curve, and then you generate it if you'd like to see the values first. So here you can see its values. You can use these to decide if you want to use that one, or you can use the 5-parameter logistics and generate that and compare the two values. So this one and this one, you can see that they have the same residual sum of squares, so the error isn't going to be compounded too much on either one. So we'll just use the one I generated earlier. If you did like this file, you just save it and it would be added to this list, which is alphabetically sorted. And so we'll go back to the analysis tab, and you can select quantitative. We'll keep the export type at CSV. The threshold is grayed out since it's not relevant in a quantitative, and you can select what kinds of plots you'd like. So we do photon counts versus concentration in our demonstration here. Once you've got all that set up, you can do choose file, select the file you'd like to test your data on. You import, once the data is imported, you select your S-curve that you'd like to test against, and then click on the file that you'd like to apply the S-curve to, and then you'll see a nice S-curve with all the data plotted to it. And once you have this data and able to visualize it, you'll be able to export this, and this will generate a file for you. And once it's downloaded, you can open it up, and once it'll show you the file name here, you can see the S-curve attributes that we set. So we use the demo txt for our S-curve. These are the control values that were from the S-curve, and you can see the generated parameters. There's 4 here because this particular file is the 4-parameter logistics. If you pick the 5, there'd be another value right here, as well as there. And these are the standard deviations of those, and then the residual sum of squares. And then you can see the data from the array tape, and then you'll have a qualitative results matrix that you can use to see all the data. And then we also have a list of all the data. And so, you'll have your well location, your photon counts, and your concentration, since that's what we plotted against. And that will be the